professor from Haifa University, but I want to put it into a context that we already know about, which is that when the human brain deals with quantity at all, there are at least two ways that a human brain can deal with it. One of them is literally counting, literally numerical. It's something that's, let's say that for 0, 1, 2, 3, 4, we'll hear about the archerfish later, but for 0, 1, 2, 3, 4, it's not counting. It's making a sudden categorical judgment. There's a pattern when you see one thing, two things, three things, maybe four things. It's a pattern that's related to George Miller's 1956 paper. Are you familiar with this, Tali? The magical number 7 ± 2.
Oh, yeah.
Okay, so that estimate was wrong. It was probably more like 3 ± 1. But there are quantities, perceptual quantities, that the human brain recognizes suddenly. Some people call it subitizing, sort of. And that's a way of perceiving quantity. Another way is to count. And we have a capacity to count. It's a iterative recursive capacity. And then there's another way, and perhaps I'm kind of curious whether Tali leans towards the very first, which is subitizing, the second, which is counting, or the third, which is just magnitude estimation. It's not really about numbers, it's about quantity. And with that, I open the floor to Tali. Tali has kindly agreed to answer questions. I'll be watching the chat, and I'll transfer the questions to Tali, and she can decide whether she's ready to answer right away or she wants to defer it. Tali.
Thank you. So I'm really, really happy that due to COVID, I can talk to you all without traveling for 12 hours or more. So I'm very happy to be here. Thank you, Stephen, for the invitation. I'm just going to send you a table that I think will help you during my talk. And let me just share my screen. Can I share my screen? Yeah. Okay. So I'm going to talk about what do we process when we process magnitude. So Stephen, to answer your question, I do think that we can count and we can estimate, but it really depends on the circumstances, and it's not as black and white and as it was previously suggested. And please feel free to jump in during the talk. So let's say that this is Joy, and Joy is having really hard time studying math in school. And it's a shame because studies are showing that doing well in math in school can predict better physical health, mental health, better chances of finding a job with higher pay and better socioeconomical status. So in the large picture, my main goal is to find out how the brain learns math. So I can take an evidence-based approach for designing assessment tools to assess math learning difficulties and better ways of teaching math. Now this asterisk is reminding me and now you that I'm taking baby steps. So none of what I'm going to show you today is something that can be applicable to schools right now, but this is definitely something that I keep in mind during my studies. So today, I'm going to give you some background about one of the most debatable topics in numerical cognition. Do we process number, meaning the number of items in a set, or do we process magnitude, such as total surface area, density, and so on? Then I'm going to talk about magnitude integration, about combination of all available visual cues to provide an estimate. And then I'm going to dive in deeper into continuous magnitudes in between themselves. So let's start. When one of the basic questions is what are the building blocks of mathematics abilities? And when we want to study the building blocks, a popular way is study developmental studies, beginning with newborns, even 72-hour-old newborns, and studies with animals. And a popular method with this kind of population is the non-symbolic number comparison task, because it doesn't require any language or any formal knowledge. So here are some examples. This mouse, for example, needs to respond to press on a lever X times according to the number of flashes of light this is. This is not number comparison, okay? This is just to show you that there are other methods as well. Birds are taught to discriminate and choose the group with the larger items. Fish are taught to swim towards where the more items are, and so does chicks. So you see that this is number comparison is not the only method, but it's definitely a very popular one. And this kind of studies led to the ANS theory, that stands for the approximate number system theory, that focuses on the discrete property, or the number of items in a set. And here are some outcomes of this theory. This is from a model of Chinook and Dehane from 1983. And what you see here is the retinal output we see. They, according to their theory, we map the special locations of each item. Notice that we ignore the physical sizes, for example, and then it goes into a numerosity detector and we get an output. Now the output is not always exact, it depends on the quantity. Because another thing is that the approximate number system or quantities, no matter of what, it can be pitch of sound, it can be a number of the size of objects, brightness, and so on. They are all mapped into this mental number line that is a logarithmic line. So the larger magnitudes are more condensed and hence less discriminable. So if, for example, here you have 20, the output can be somewhere between, for example, 15 and 25. Another thing is that we have to separate core systems. Core system 1 to represent large numbers, and then performance is ratio dependent. Namely, if the ratio is the smaller divided by the larger quantity, 0.2, for example, can be 20 versus 100. 0.8 would be 80 versus 100. So performance is much more difficult as you get closer to 1. It's ratio dependent only for large numbers, not for small numbers. Now, one of the reasons that the ANS is so popular is that it was found to be correlated with math abilities, and even suggested that this is the basic ability that we need to study later math. So this is a study by Halberda et al. From 2008, highly citable paper, where he asked 14-year-old participants to decide whether they see more blue or more yellow dots. And he changed the ratios between them and to see what is the most subtle difference participants can still distinguish. And this is the Weber fraction. The lower it is, the better you are at discriminating smaller and smaller differences. And on the y-axis, you see the score in a calculation test. So you see this correlation. It's important to note that other studies tried to replicate it. Not everyone succeeded, and those who did got a medium to a small size effect. However, there are several challenges to the ANS theory. First is the correlation problem. This is what you will see a typical stimuli in the number comparison task. So you see that the number changes, but not only the number of dots changes, but the physical size too. You can't get into a situation where you have a different number of items, but all their continuous magnitudes stay the same. And when I'm talking about continuous magnitudes, I'll be talking about 5, the density of the dots, the convex hull, which is if you connect all the dots in the periphery, what would be the area of the dots and the space between them, the total surface area of the dots, their total circumference, and their average diameter, if inside every group you have different size of dots. And usually this continuous magnitude in everyday life correlate with number. It means that, for example, the more apples I buy, the more heavier they will be, the more space they will take in the drawer and so on, compared to fewer apples. So if it's usually correlates with the environment, why do we ignore it? The second challenge is the computational demands or the adaptivity. Some studies have suggested that it's less demanding computationally to just compare the area, for example, than the number. And it can be very important. Think about it from a perspective of an animal. So say that this gerbil needs to decide which pieces of food to take, and he needs to do it very, very quickly because he is now outside exposed to predators. Would it be more adaptive to do a fast estimation maybe of the height of the pile or the density instead of estimating the number? Because he needs to make a very quick decision. The third challenge has to do with the different environmental demands. So maybe sometimes you do need to be very accurate and count and rely on number. For example, if you're baking a cake and you need exactly 3 or 5 or 8 eggs, you want to be accurate. The same thing if you go on a field trip with 10 children, you want to go back exactly with 10 children, not something that looks about 10. However, if you need to decide which line to wait in, instead of counting the people, maybe you can just estimate where there are more by the line, by the length of the line that they make.
Tali, would you accept a question at this point?
Of course.
What role does the distinction between a relative and absolute judgment play in what you're saying now? Magnitude estimation is relative, whereas now you're talking about, when you're talking about exactness, you're talking about absolute judgments.
Yes. So sometimes maybe there is logic in being very precise, but sometimes it's not. And it doesn't yet clear what we do when we compare. For example, if you take these two groups of dots, so some would say that we compare between them and then decide, while others would say we estimate how many there are here, we estimate how many there are here, and then we compare them.
And the comparison is a relative judgment. Is this more or is that more? And the how many there are, I don't know what it means, but when you say how many there are is an absolute judgment. It's basically asking to count.
Yes, but you can also ask to estimate how many. And it's very, very interesting because if you're doing, for example, if you're asking people to place a quantity on a line, you say, okay, imagine that the leftmost point is 0 and the rightmost point is 10. Where would you put this quantity that I'm showing you for 100 milliseconds? So you need to estimate. At that, in this task, people usually estimate, and when they estimate smaller items to be more, and larger items to be less. But in comparison tasks, you get the opposite. Like here, you assume that the physically larger dots will be more. So this is very nice because you see a distinction between estimation, which is more absolute, and comparison that is more relative.
Thank you.
Okay. There are also direct empirical evidence that support the role that continuous magnitudes have in a non-symbolic number comparison task. Kelly Mix and colleagues wrote a very nice paper in 2002 that review all the developmental studies that demonstrate that the ability to perceive quantity is innate and explain it by continuous magnitudes, because they weren't controlled, so they were a possible alternative explanation to the results. Now, researchers are well aware of the correlation between continuous magnitudes and number, and this is why they're trying to break it in the experiment. So imagine that you are asked where there are more dots. And in 50% of the trials, you see the larger number of dots and the larger continuous magnitudes all in the same group. So these are positively correlated. There is a positive correlation between continuous magnitudes and number. But in 50% of the trials will be incongruent. So the larger number would be in one group and the larger continuous magnitudes would be in another group. And this is a negative correlation. So if half of the trials are congruent and half are incongruent, in total, you don't have any correlation. And this is where some researchers are saying that because the continuous magnitudes are no longer helpful cue of numerosity, they can't be used as a cue, as a clue to the right quantity, participants will just ignore them. And what we will measure is a pure number sense. But what happens when you separate congruent and incongruent trials? Then you get faster response times and higher accuracy rates for congruent than for incongruent trials. Now, this gap in response time or performance is called the congruity effect. And this is the hallmark of automatic processing of the irrelevant dimension, the continuous magnitudes. So we can do this task, but we need to exert cognitive control to do that. So you don't only measure number sense, you also measure cognitive control. And there are very nice studies demonstrating it with fMRI or ERP that we do activate cognitive control in incongruent trials. Based on such studies and many more, my colleagues and I suggested the AMS theory, the approximate magnitude theory. I won't go into the entire model. I'll just tell you that it assumes that we are not able to discriminate quantities from the moment that we are born. What we do instead is to compare very crude continuous magnitudes. Then we learn the correlation that we talked about between a quantity or number and non-numerical magnitudes, continuous magnitudes. And with cognitive control, we are able to break this correlation and use continuous magnitudes when it's appropriate and number when it's appropriate. Okay, so now after hopefully I convinced you that continuous magnitudes matter, I want to move on and talk about magnitude integration. As I already said, there is a natural correlation between continuous magnitudes and number. Usually they positively correlate. So given this natural correlation between continuous magnitudes, I wanted to see whether there is an area in the brain, a network in the brain that can integrate these continuous magnitudes. This is a study I did in 2016 with Daniel and Sari. It was an fMRI study. We typically developed adults, and we had two different tasks. The quantity comparison task, we had four different levels of congruity. So if you remember before, I showed you that in congruent trials, all the continuous magnitudes were positively correlated, and in incongruent, they were all negatively correlated. So here, we have different levels of congruity. In congruity level 1, only 1 out of 5 continuous magnitudes is positively correlated with number. And in congruent level 4, 4 out of 5 continuous magnitudes are positively correlated with number. Now the task here was to choose the group with the larger amount of dots. You can say that congruity level 4 would be the easiest, right? Any questions, by the way? Okay. Next, we had a control task with brightness comparison. We wanted to have a type of magnitude to compare in a 2 first choice task. So here we asked which rectangle is darker, and we wanted to match the difficulty. So the ratio of 0.4 between the luminances was as difficult as congruity level 4, and the ratios of 0.85 was as difficult as congruity level 1. These are the fMRI results that we got. So what you see here is an area that was working differently depending on the different task and the difficulty level. I hope you can see this yellow dot. It's the same dot, but you see it in different perspectives. So sagittal view, it's as if you're looking at the brain from here, from different depth. Coronal is if you're looking at it from this perspective, and transversal is from here. So it's the same spot from different point of views. And this area is the right inferior frontal gyrus. You see that its activity in the numerosity comparison task decreased or increased when there are more clues, there are more continuous magnitudes to integrate. In difficulty level 1, which was the easiest, you had 4 continuous magnitudes you can integrate with quantity. And this is when the activation was higher, and it decreased until difficulty level 4 when you have only 1 continuous magnitude that you can integrate with number.
Could I ask another question?
Yes.
May I ask another question? What exactly was the instruction to the participants? What were they asked to do?
They were asked to choose the group containing the larger number of dots.
So it really is, it's strictly quantity, and what you're inferring is what did they use to base their quantity judgment on?
Exactly. Now that's the problem with people, right? They always ask you for instructions. And this is, by the way, where the archerfish comes in. So I just wanted to say that in the brightness task, what you see, the slope is actually 0. So we see that when you have nothing to integrate, the activity stays the same and is not influenced by difficulty. So it's not only difficulty, it's the combination of quantities. Now, as I said, we need to ask people things. So we don't know what spontaneously they want. This is why we ask if we will use the same stimuli as before that I just shown you with the 4 congruity levels. Will an untrained animal spontaneously choose the group of dots according to its quantity, the continuous magnitudes, or the combination of all of these visual cues? So for that, we use the archerfish. So the archerfish hunts insects that live above the water level. It can hunt them by shooting a jet of water at them, and then they drop. It can predict where they're going to drop and eat. So this was our animal model for several reasons. First, the hunting method. Second, their brain structure. It's very, very simple. You can see optic tactum and telencephalon, not a lot more than that. Fish and humans diverged a long time ago in evolution. So if you see something that is behaviorally similar for fish and humans, it either means that it was preserved throughout evolution or was important enough to be developed independently in different species. Another thing is that these fish are highly trainable. We have them in the lab, and we can train them to shoot water, just like you saw in the video, but on a computer screen that's showing artificial targets that we present them. So it can be an asterisk, a number, or whatever we want. And then you get the parallel of a person doing a forced choice task by clicking on the keyboard. Importantly for us, fish don't ask questions. We don't need to give them any instructions, and we can know, and we can see if we don't train them and reward them regardless of their choice, we can see a spontaneous preference.
So, okay, but let me, still, one can say, you didn't give them instructions, they have a preference, but a preference for what?
Exactly. This was our question. A preference for what?
But here it's going to be, the answer to the question is going to be what task they thought they were doing.
I don't know if they care about what task they are doing.
Well,
I mean,
not for you, not for the experimenter, but when they're in this situation and they have a choice, there's something that they're doing when they're making the choice. And the question is, what are they doing?
Exactly. So these are the most neutral conditions that we were thinking about, trying to somehow mimic what they have in nature. So what are they doing in nature if they have two different groups of insects standing on different foliage? What is guiding them then? Okay. So this is the general setup of a fish experiment. We have one fish per fish tank with a computer screen above. It's rested on a clear glass shelf, and it's hooked to a computer so we can present the stimuli. We record the experiment with fast video cameras that are synced, so we can extract response times if we want to. This is an example of one fish in the lab. You can see the jet of water. And this is just an illustration of the stimuli. This is how the specific experiment that I'm telling you about went. So the fish saw 3 flashes of these boxes that were in either one of the corners of the screen, indicating where the stimuli are going to appear. We never do it in the same spot because we don't want the fish to always select one specific spot regardless of the stimuli. And then after a short blank screen, they see these 2 groups of dots, and they have 15 seconds to decide which group they're going to shoot water at. Once they do it, we give them a reward regardless of what they answered. And then they have 10 seconds to eat. We have time to clear the screen, wipe the water, and get ready for the new trial. We had 7 fish. Each fish had 24 sessions of 48 trials each. When they saw exactly the same stimuli with the 4 congruity levels I've shown you in the fMRI study. These are the results. So on the x-axis, you have the different congruity levels. You can see examples here. And on the y-axis, you have the chances, the proportion of selecting the group with the larger quantity, with the larger number of dots. And each colored line is a fish, and the black one is the mean of all the fish. 50% is chance level. So what you can see is that for congruity levels 1, 2, and 3, the fish preferred the group containing less dots and larger continuous magnitudes. But in congruity level 4, it switched. So they preferred the group with the larger quantity and larger continuous magnitudes. Now in congruity level 3 and 2, for example, we had different two continuous magnitudes that were positively correlated with number. And we tried to see if there are differences, and there weren't any. So it's not the specific continuous magnitude that made a response, but rather the number of continuous magnitudes correlated with number. Okay. So moving on. So far we talked, yeah.
Tali, just a comment. Is that the end of the archerfish story?
For that one, yes. But if you want, I can
No. I don't. I was afraid for a moment when you showed the archerfish brain that you were going to hurt them. Okay.
No. What we do is strictly behavioral.
Okay.
And they actually enjoy it. Sometimes you see we need to cover the aquariums to the sides because they try to answer it when it's not their turn.
Archerfish are little geniuses. There are many people are interested in archerfish cognition.
They are, and I think that they are a great animal model. The only restriction is that we get them, they can't be raised in the lab. So we get them from importers, and we don't know whether they are males or females. We don't know how old they are. We don't know if they're sick or not. And so we have all these unknowns that we're dealing with, and I really, really hope that someone will be able to breed them in a lab, and then I think it will be the new zebrafish.
What did the biologists say about why it's so difficult to breed them in the lab?
Apparently, my husband can tell you more about it. He's a biologist. Apparently, many animals have very, very specific times when they breed, or very specific condition. It can be the length of the day, or funny things like flamingos, for example, apparently need mirrors to procreate. So I think that and if you don't know which fish is male and which fish is female, or if they're constantly male or female, how do you know who to put with whom? So it's true.
It's true. I mean, statistically, in the end, you should be able to approximate it, even if you don't know what the gender is. But okay, very interesting. Thank you.
Yeah. Okay, so far we saw that the set of items can be characterized by either the quantity, the number of items in a set, or continuous magnitude. Until up to about 2 decades ago, the focus was on quantity, the non-symbolic number. In recent decades, now the role of continuous magnitude is acknowledged and studied more, but we still see them as a group, as continuous magnitudes. But they're made up of different things like density, total surface area, etc. So what happens if we study them individually? This is a study led by Mazal Ezra, one of my master's students. And what we asked in this study are 2 questions. First, in a cognitive magnitude comparison task, just like the one I showed you with the dots, but this time we asked about continuous magnitudes. We wanted to see whether participants can selectively attend to a specific continuous magnitude according to our task instructions. So they can they focus on just the total surface area or just the convex hull, and how it will affect performance. And the second question is, under which conditions quantity will be processed automatically, and what would characterize this automatic processing? And I want to take a moment to focus on this question and explain its significance. I've shown you before the congruity effect, and we talked about how this is the hallmark of automatic processing. But sometimes the congruity effect can be broken down into 2 components. To do that, you need a neutral condition. So imagine that you see these 2 groups of dots, but you are asked about the total surface area, where you see a larger total surface area. In that condition, In this question, you can have a neutral condition when you keep the number of dots equal and only change the continuous magnitudes. And then you see this pattern of response times. Usually congruent is the fastest, incongruent is the slowest, and neutral is somewhere in the middle. So facilitation is the time difference between neutral and congruent, and it tells you how much having another clue going in the same direction as the correct answer facilitated, helped the performance. On the other hand, you have interference by how much having a confusing dimension, anti-correlated one that goes against the right answer, hinder your performance. And these congruent components are not often studied in non-symbolic stimuli, but when we do study them in symbolic stimuli, we see very interesting things. This is an example for a study by Orly Rubinstein and Avishai Henik from 2005. They asked adults, typically developed adults or adults with developmental dyscalculia, which is a learning difficulty specific to math, to say which number is physically larger. So here in congruent trials, this will be the 5. It's congruent because 5 is numerically and physically larger. In neutral, you only play with the relevant dimension. And in incongruent, 5 is the correct response, but it's numerically larger and physically smaller, and this is why it's incongruent. And they check facilitation and interference. So you can see that in typically developed adults, you have both interference and facilitation. But in participants with developmental dyscalculia, there was only interference and no facilitation. Now, according to Posner, facilitation is a sign of automatic processing of the irrelevant dimension, that you see that it really helps your performance. And people with developmental dyscalculia don't enjoy it. By the way, this is also the typical pattern you see in typically developed 7-year-olds. And interference is a sign of more attentional processing. So it can show you how separating the congruity components can shed more light on the different processes that's going on. Back to our study, we had 3 groups of participants. One group was asked about where the average diameter of the dots is different, because the dots were in different physical sizes in each group. One group was asked about the convex hull, and one group was asked about the total surface area. So average diameter, AD stands for average diameter, CH for convex hull, and TS for total surface. We also manipulated the stimuli. So largely we had congruent, incongruent, and neutral trials. Okay, congruent, all the continuous magnitudes were positively correlated with number. Incongruent, all the continuous magnitudes were negatively correlated with number. And in neutral, we had exactly the same number. The groups contained between 10 and 40 dots, and the ratio between the number of dots ranged between 0.4 and 0.6. I'll be getting to why very soon. And we manipulated the stimuli differently. How? In the average diameter condition, the ratio between the average diameter was identical to the ratio between the quantities, between 0.4 and 0.6. In the convex hull condition, the ratio between the convex hulls was between 0.4 and 0.6. And in the total surface area, the total surface area was a range between 0.4 and 0.6. Why it's important? First, these are the results. This is the table I sent you. And you want me to send it again because people who weren't here.
We have it in the messages, so it's there. I was able to open it, and anybody can open it, I think, from the message.
Okay, but so sometimes people that join late can't see the chats from before, so I just joined. Okay, I just attached it again.
Okay.
So what you see here is the different manipulations we did and how the ratios between the total surface, the average diameter, and the convex cells were affected. In the average diameter manipulation, you can see that the average diameter ranged between 0.4 and 0.6. And in the other things we can't control, so we just recorded them. You can see that in the total surface area, the differences were huge. A difference of 0.2 is very distinguishable, very hard, difficult to miss. But if you take a look at the convex hull condition, it's more difficult. You see there are things that there are stimuli that are 0.99 similar, which is very, very, very subtle difference. Okay, and you can see that the convex hull here was between 0.4 and 0.6, and the total surface here was between 0.4 and 0.6. Keep this in mind when we're going to look at the results. Now, why different saliences are so important to us? Let's demonstrate with an experiment that uses symbolic numbers. If I'm asking you which number is larger in value, here it's 6, it's very easy. Here it's also 6. Here it was easier than here. Why? So in 2013, my colleagues and I had this experiment when we showed these pairs of numbers, single-digit numbers, and we manipulated the numerical ratios. We had 8 different numerical ratios and 8 different physical ratios, congruent and incongruent trials, so it was 8 by 8 by 2 number of conditions, and we wanted to see how the size of the ratio of the relevant and the irrelevant dimension affects the congruity effect. So you see here that numerical ratio was the relevant dimension. This is what I asked about. And the physical ratio was the irrelevant dimension. Now, if you look at the difficulty in the numerical ratio, the relevant one, the difficulty increased with the increase of ratio. So here, instead of 2 to 6, if we had to compare 8 and 9, it would be much more difficult. But what happens in the irrelevant dimension? It's exactly the opposite. So here, the most difficult condition was when the physical ratio was closer to 0. Why? Because then the difference is very obvious and it's really difficult to ignore such an obvious effect. So this is why sometimes you can get a very high congruity effect, and sometimes you can get no congruity effect at all. To summarize, we understand now that the congruity effect is affected by both the saliency of the relevant and the irrelevant dimension. And in the current work, to simplify, we kept the ratio between the quantities, that is the irrelevant dimension, constant between 0.4 and 0.6. Why? Because we know from previous studies that this ratio is salient enough to be processed even when it's not relevant to the task and we wanted to check congruity effects. This is how a typical trial looked like: a blank screen, a fixation in the center of the screen, and after a while, a group of dots appear and you have 3 seconds to decide, according to the task instructions you got, which of the 2 groups of dots you select. So the question type was different from different participants, but all the participants, so all the manipulation and every manipulation, including all 3 congruity conditions. So these are the results. First, we wanted to see if participants are able to focus on one relevant continuous magnitude. So what you see here is a different plot for every question. So this was the average diameter question, the convex hull question, and the total surface area question. On the y-axis, you see the accuracy. 0.5 is chance level. This is the red line. And the third triplet is the average diameter manipulation, followed by the convex hull manipulation and the total surface manipulation in congruent, neutral, and incongruent trials. So if you take a general look you see that performance was usually above chance level. Accuracy was very high, except for here, when most participants were at chance level. And why is that? To do that, we need to take a look at the table I've just shown you. Remember that in incongruent trials, the ratio between the convex hull, which is the continuous magnitude that participants were able to focus on, it was hardly distinguishable. And this is why they just had to guess. But in the neutral and in the congruent trials, when number was not there to confuse them, they performed fairly well. Now, notice that here they saw exactly the same stimuli, but they had to attend to the total surface area, and here to the average diameter. So it's not like they couldn't avoid the convex hull, but they chose to focus on it when it was the relevant dimension to the task. So even though all of the dimensions I told you about, total surface area and average diameter and convex hull are all correlated, we are still able to extract one of them and focus on it. Second thing we saw is that the question type itself influences efficiency. Efficiency is response time divided by accuracy. The lower it is, the more efficient you are. So what you see here is the different manipulations. So all the average diameters are exactly the same stimuli participants saw, but under different question types, under different instructions. And you see how these instructions affected their efficiency. This blue bar here is the convex hull when it was hardly distinguishable. And this is why you see that the efficiency was really, really poor. And you can see it affected differently the different conditions. The second question was about the congruity components. So facilitation, the time difference between neutral and congruent condition, and interference, the time difference between, or efficiency difference between incongruent and neutral trials. And for every manipulation and every question type or instruction type, we wanted to see whether facilitation and interference existed, whether they were significantly different than zero. These are the p-values. So you see that in the average diameter manipulation, we always have facilitation interference. In the total surface manipulation, we had only interference, and in the convex hull, it depended. Now we wanted to see whether these facilitations that are all significant are different in size. And the same goes for these facilitation and these interferences. So this is the next plot I'm going to show you. As you can see here, the facilitation was indeed different in size, depending on the different questions that we ask participants, different continuous dimension we ask them to attend to. And the same goes for interference. Another thing we wanted to see is whether facilitation and interference are different in size from one another, because again, facilitation and interference indicate different things. So this is what we see here for every question type. The facilitation and the interference, we see that we always have larger effect for interference than for facilitation. Now, according to Posner, I remind you that facilitation is the hallmark of automatic processing, while interference is the hallmark of attentional processing. So here it seems that we have more attentional processing of number than automatic processing. So just to conclude this study, adult participants are indeed able to focus their attention on a single continuous magnitude. The saliency of the relevant dimension affected the processing of magnitudes, and this finding supports the approximate magnitude system theory, suggesting that the saliency of quantities and other magnitudes depends on top-down factors such as the task instructions and bottom-up factors such as the saliency of the different magnitudes.
This might be a good point for a pause before you give your final take-home message. There was a question earlier after the archerfish from Nua Atlas. The question is, have you done it on, two questions actually, have you done it on other organisms? For example, she mentioned mammals like dogs. And the second thing is about training. I mean, and this is true about, this question is valid both for animals and for humans. I should say for human animals and non-human animals. You tell them to pay attention to a dimension, but what about training?
Yes. So whether it was done on other animals, spontaneous discrimination, yes, it has been done. And you see that when there was actually a group that in 2012 did this similar study with angelfish. They wanted to see which plate of foods they would choose. And in 2012, they didn't control continuous magnitudes, and then they decided it's quantity. In 2020, they repeated the experiment, but this time they controlled the size of the food, and they saw that it's influencing the decision making. And there was even a condition when they had the different number of items, but their physical size was the same, and then performance wasn't chance level. So yes, it happens with other animals too. Once you can control, now not all the time we can get these very accurate results like we see here, because the archerfish allow us to do many repeats and get very good results, very good data. And for example, with bees, you can only do a single trial or something like that.
So that answers the comparative question, but what about the question about influence of training? And by training, what's meant not just the training of the archerfish that they get rewards after, etc. But what if the rewards were favoring one dimension over another?
Yes, so this is a study that we are planning to do. But there are studies with different sorts of fish, different kinds of fish, that do show that you can train them to use symbolic numerical magnitudes, but it's easier to train them to discriminate between continuous magnitudes.
Fine. So another species is human. What about training in humans?
Yeah. So humans, you can tell people to select the smallest number or the largest number, but you don't know exactly if this is what they're going to go by, because as I said, it's very difficult to control the influence of other continuous magnitudes. So some will try to get cute and say, okay, so I will always select the smallest magnitude, for example, or something like that. So in humans, you don't need to train, you just give instructions.
Yeah, instructions is one thing, and then you find out spontaneously what they do when you tell them to pay attention to this or pay attention to that. But training is another question. I mean,
training to do what?
You could favor some aspects. It doesn't even have to be particularly one. You could favor ratios and things like that, and see how, in the same way that you're interested in how many more mistakes they make under this condition or those conditions, you can also find out if you've been favoring something, what does it do to the overall effect? What's behind this is, of course, this is a cognitive informatics seminar, and we're going to be interested, although you don't do it, we're going to be interested in models for this. The underlying process, including learning.
So one thing I can say about it is, I really don't know enough about, for example, computational modeling, But there is one study I know of, it was done by someone who was with me during the PhD, Gali Katz. She was from computer sciences, and she tried to tackle this question of continuous magnitudes and quantities by using evolutionary algorithms. So what she did is she trained one algorithm first to discriminate between continuous magnitudes, and then discriminate between quantities, and compared it to an algorithm she trained right away to discriminate between quantities, between numbers. And she saw that the learning of discrimination between numbers for the algorithm that was first taught to discriminate between continuous magnitudes was much shorter and efficient than the algorithm that only learned from scratch to discriminate between quantities, if that answers your question.
Well, it would be nice to know what that implies. You know, one of the interests in cognitive informatics is in producing software that will help children learn math, okay? And for that sort of thing, it's good to know what their starting point is, what their biases are, and what scope there is for changing them with various kinds of schedules.
So one thing that I really wanted to do is to build, I didn't call it a software, but a training, but it's the same, I think. It will be even nicer with the software, is to try and train. I'll start earlier. So remember that at the beginning, I've shown that they think that there is a correlation between how well you're able to differentiate between quantities and how good you are in calculations. So there are many studies that are trying to train the ANS to try and you know, by giving feedback and make participants better at this perception, and they are getting better, but it doesn't affect mathematics.
What do you mean?
It means that their performance in the number acuity task gets better. They can discriminate between finer and finer differences, but it doesn't affect their math abilities. It doesn't.
Oh, I see. Yeah, and this is in children of what age?
It's been done in different ages. I think that usually elementary schools.
So you think that the underlying ability is pretty rigid and not plastic?
No, I think that there is something called the symbol grounding problem. We still don't know where our symbolic understanding of number is coming from. One theory is that we first learn these non-symbolic quantities, and then we match the symbols with the quantity. But sometimes it doesn't make sense, because you see, for example, that 4-year-olds are better in comparing symbolic numbers than non-symbolic numbers. So how can they be these spaces if they are less proficient in that? Sometimes you see that when children improve in discriminating between symbolic numbers, saying which one is larger, the symbol 4 or the symbol 6, numerically, then they become better in comparing quantities. So we are not sure that these two are related. One thing that we do find, this is a study of Bogdan and Ansari from 2015. They have done this non-symbolic number comparison task with congruent and incongruent trials on elementary school children with and without dyscalculia, learning difficulty in math. And they've shown that it's only in the incongruent condition that children with dyscalculia are different from children without dyscalculia. So maybe it's not a problem of perception of quantity at all. Maybe it's a problem of inhibitory control. of inhibiting the effect of the continuous magnitudes. Maybe it's a problem, and this is what I think of understanding the initial understanding of the correlation between number and continuous magnitudes, and then the ability to break it. And this is where my studies are going. So I want to see whether participants, for example, children with better facilitation abilities are better in math. And then whether if we will train facilitation, we will see any improvement in math abilities. So I'm not sure that the non-symbolic quantity system is the basis for the symbolic system we see. Maybe it just develops independently.
Okay, I'll let you do your conclusions now. It shouldn't be a surprise to you that the symbol grounding problem is one of the themes of this entire series of seminars.
That's good.
Okay, go ahead, please, Tali.
Okay. So we saw that since discrete number and continuous magnitudes all affect performance, we should never assume that we can control their influence, and we should always study their possible influence, even if it's not the goal of the study. We saw that it's also important to study continuous magnitudes, not only as a group, but as separate magnitudes. Now, it's been done, for example, in perception. There are many studies about perception of density, for example, and it can add more knowledge to what we know. Camilla Gilmore, for example, has nice studies about how convex Hull can affect performance differently in different stages of development. And it can also just being aware of the differences between different continuous magnitudes can help to reduce the noise in the data. Because usually when we talk about continuous magnitude as a whole, we try to give very vague instructions, like where you see more yellow area, if your dots are yellow. And then we take the chance that each participant is attending to a different dimension. I once asked participants after a task that was meant to focus on number, according to what they decided. And some told me total surface area and some density, and some even gave me this formula that they think that they used. So if different continuous magnitudes have different saliency, we can get very noisy results. Another question is whether different saliency of different the saliency of different magnitudes can change across development, or can be different in dyscalculia. And as I told you before, computational models can provide important insights in this field. So that's it. I'll be very happy to answer questions.
Maybe you could unshare and then we can have general questions.
Yeah.
Thank you very much.
Thank you.
Okay, so the questions are open to everybody, and vous pouvez poser la question en français, on peut la traduire. Donc, questions in both languages are welcome. Go ahead. If not, I always have a few more before. I won't let Tali get away without some more questions.
I just want to say that I did a small experiment with my dog, and I can just share with you the link to the little YouTube video I did about it, to explain about the ANS versus the AMS theories. But you can go ahead and ask.
Okay. Meanwhile, let me set it off by asking, could everybody, of course, will be thinking about two authors. Anybody who's thought about this problem will be thinking about two authors who and two researchers who are behind it all. What is, in a more Alan Alda plain sense, what is the relation of your findings to the original findings of Piaget about conservation?
Oh, This
Everybody
is
must
very,
ask,
very.
everybody asks you that question, but you have to still make it explicit.
I love this question because if I'll go back to, can I share again?
Yes.
Okay. If I go back to the model I suggested, there we go. So I'm talking about first over applying the correlation between number and non-numerical magnitude. And this is very similar to what we see in Piaget's number conservation task. In this task, do you all know it, or should I explain very
Let me
briefly?
suggest that you should do an Alan Alda approach now to everybody.
Okay.
There's a large proportion of people whose main competence is in computer science and not in cognitive psychology.
Great. So let's do this live. Let me improvise in the spirit of Elenada. Okay, so let's say we have this is what a child sees, okay? Oops. 2 rows of coins. And the experimenter asks him, does this row has more coins? Does this row has more coins? Or are they the same? And I'm talking about kids before the age of 5. So some of them will even count and will say the same. And then right in front of the child, the experimenter space out one of the rows. And then repeats the question. And then without even thinking, the child would say, can you guess? This row has more quarters.
Yeah.
Why? Because it's bigger. And this is an example of over-applying the correlation, because here, technically, you can have more coins and they take more space. And usually, if you had more coins, they would take more space. And this is what happens when you first learn the correlation. Later, when you develop inhibition abilities, you know when it's appropriate to use it and when you need to ignore it. Now, I know that this task is very debatable because, for example, if you do it with M&Ms instead of coins, children know exactly how many even when you space it out. So it can be dependent on the way Piaget asked the questions or the stimuli used. But LaRoche and his group in 2009 did a similar study with adults in fMRI. And of course, adults knew exactly the question, but they activated more areas of cognitive control under this, what I would call an incongruent condition. So does this answer your question?
Yes, and are the pre-conservation children like the archerfish, or are the archerfish more like the post-conservation?
I don't know, because I didn't ask the archerfish if it prefers numbers. This is actually on the plan. But what I can tell you about the archerfish is that I've tried to teach it symbolic numbers, the order of symbolic numbers. So its stimuli was the symbolic numbers 1 through 4. And I tried to actually train it, for example, to shoot water at the larger number. And between 1 and 4, it worked after a lot of training. When I'm showing the fish in the training phase only consecutive numbers, so 1, 2, 2, 3, 3, and 4, it performed above chance level. And in the test phase, again, I'm showing only non-consecutive pairs like 1, 3, 2, and 4. And the performance was again above chance level. And it also had a size congruity effect, like you saw with the 3 and the 5 that were different in sizes. But when I was trying to repeat the study with 5 digits, I saw that he wasn't able, they weren't able to learn anything above chance level. So what I saw was probably an end effect. They learned the edges, the anchors, but not the middle. So I don't know if a fish has transitivity, which according to Piaget is very, very basic and required for these jumps in cognitive abilities.
Thank you very much. Could you do the same thing, but this time not with Piaget? With Piaget, you were reasonable to, it was reasonable to assume that everybody here knows what Piaget says or understands. Another writer in this area that you're familiar with is Gallistel. And I wanted to know how your work bears on his, but for that, could you please also give everybody an idea of what Gallistel says?
So you're talking about the numerosity detector?
Yeah, and his book, there's a whole book on this, so yeah.
Okay, so what is basically saying, if I understand you correctly, is that we are representing numbers analogically, but it's noisy. So he's comparing that to pouring a water into a beaker. And if you're pouring a lot of water, then it takes the water a while to set on the right scale. And this is when you don't exactly know what the quantity is. So I think Gallistel talks more about estimation, and I don't know how much of his studies are concerning the role of continuous magnitudes and comparisons. But correct me if I'm wrong.
Well, he introduces, his notion is that at low ends of the quantitative continuum, it's another process. Continuous magnitudes only happen when you exceed the threshold for the other, and I would say that, although it's not exactly correct, but the lower end process is an absolute one. You can train archerfish, etc., to literally to make an arbitrary response for when there's one thing, two things, three things, four things, and so they can depend.
Yeah, we actually did it.
Yeah, you did. Okay, so that is an absolute task. You're basically saying if you give a relative task, but where you have a participant who can count, then obviously they're going to count both of them, and the higher count gets the. But above a certain value, according to Gallistel, that's not possible, and you have no choice but to go into things like convex hull and whatever is affecting your magnitude perception.
This is what Gallistel is saying.
Yeah.
But I forgot the names of the other terms. This is the Core System 1, Core System 2 theory from 2004. They're actually saying the opposite. So they're saying that in larger quantities, we don't use continuous magnitudes, but in smaller quantities until 4, we do use them.
With what you said about logarithmic, I mean, basically psychophysics says that how can that be? I mean, you get
I know.
really fuzzy.
So, according to studies they did, they demonstrated, they show an influence of continuous magnitudes only in the lower range. But, it can be a methodological issue because, as you say, if I'm thinking about it computationally, it's very easy. It's a pattern recognition almost. Although there are arguments about this, for example, about what comes first, the ability to subitize, to make a very fast and accurate estimation of the quantity that we see, or is it just a perfection of counting? Now it's a chicken, and it sounds like a chicken and an egg problem, and because you in these kinds of tasks you need participants to say aloud how many items they see, and you can do it with pre-verbal children. But something that I am trying to do, and because of COVID, I can't do these experiments now because they require the use of eye movements. Some studies have demonstrated that you do less eye movements in the subitizing range. And you can see by, according to eye movements, when participants start to count. So this can be nice because then you don't have to, you don't need the participants to say anything. You can just let them see something and use an eye tracker to see that. So, and in addition, my colleagues and I developed an algorithm that can estimate the individual subitizing range of every participant, which is something that was always done at the group level by then, or in a method that did a gross overestimation and were not accurate. So what we're thinking about doing is try and see if we can measure the subitizing range only with eye movements, and then try to start and measure subitizing in starting with 9-month-old infants to see whether subitizing comes before or after counting.
It makes sense to think of, well, that's a good question, and but it makes sense to think that eye movements would be a cue to the fact that you're not, well, in subitizing, it's sudden. You look at the whole pattern and you see there's 3, there's 2, there's 4, there's 1.
Although some people will argue that you do count, but you do it really, really quick and really efficient because the slope is never 0.
Okay, so let's, yes, but that still fits with what you said, which is that
Yeah.
very little eye movement when you're subitizing, and it's more once you're counting. But according to Gallistel, there's 3, right? There's subitizing,
Yeah.
there's counting with some have, and then there's just plain continuous magnitude estimation.
Yes, I think it's more task and context dependent than we think. Because even in my experiments, in my enumeration experiments, you can see how flexible this range is. So some people will start guessing from 7, and some people will take the time and count even until 9 and 10. And I think it will change if you change, for example, their motivation, if you give money rewards for how accurate they are, or how long you give people time to view the experiment, the stimuli, whether they can use additional strategies such as groupitizing, which is when you take small groups, divide them and see and do like, if you can subitize within the groups and then add them up. So I think that it's more complicated than something static. I think it does exist. But again, if you need to make a split-second decision, even if you have like only 20 or 30 dots, you may have to use continuous magnitudes. Or maybe you don't need to go into the estimation of number. So it's really task-dependent and saliency dependent. Of course, that if the numerical ratio is what's obvious, you will notice this. The numerical ratio, but if the total surface area is the largest one and it's a better cue, why not use it independently of the range?
If there's no other questions, I have one last question if you're not exhausted.
No, I'm fine.
Okay. I mean, we have geometry and we have algebra and we have arithmetic. I have a feeling that in geometry, I'm sure that parallel processing and vision plays a role, but in arithmetic, it's much more likely to be serial. And the first question is, have you looked at temporal numerosity rather than spatial numerosity judgments?
I wanted to, but this is, it's very difficult because then you don't know what they remember whether they remember, for example, the surface area or the convex hull, and how exactly you control these continuous magnitudes in time.
Well, surely it's one-dimensional, but it's the same sort of thing. You did it with your dots, right? With it's by the frequency and the distance and the interval between.
So what? So exactly. So maybe I misunderstood you. Can you repeat the question? And what do you mean of temporal?
You can compare if we do your kind of experiment where the participant, whether it's an archerfish or a child or a human, says which is more, and they have to choose between two, and the sequence is temporal. Da da da.
You really present them.
Yeah.
Then you have the same number, because if the same problem of a confound because and it was done a lot with children. If you want to show more dots and fewer dots in the same sequence of time, in the same length of time, then you have to show more dots faster, and this can be a cue. So it was done a lot developmentally, also cross-model studies. When you see a video of a dog beats twice on a drum, but you hear 3 drum beats, and they see if the looking time is different than when you see 2 and hear 2 drum beats. But they all suffer from the same confounds, and that's the main issue.
Yeah, but the time domain is such that you can manipulate the length of the interval, the absolute length. You can manipulate the length of the sound, and of course that there's an interaction there. If you manipulate the length of the sound and the interval, then you have to have something has to give. Anyway, with that, I want to thank you very much, Tali. I'm sorry that the interaction was mostly with me. Usually there's more questions from others. Well, maybe I should take a pause. Est-ce qu'il y en a qui sont gênés, qui auraient une question à poser? Is there anyone that was inhibited but has questions to ask? Matthew, go ahead.
Yeah, so this is related to training, actually. I was just wondering how much practice your participants are given in that sort of final big experiment with 4 different dimensions going on.
You mean the training that we do before the study to see whether they got the results? Got the instructions right, sorry?
Yeah, so is there any period where the people can sort of practice the task and figure out how it works before you start recording their data?
Yes. What we did in the last study that I've shown with the 3 different instructions is we gave a practice and started the experiment when participants answered 10 trials correctly in a row. And we limited the number of trials to 90, but no one reached that sort of that limit. So usually it's done pretty quickly, as long as they understood, for example, what the total area is or what convex hull is. They're able to do it very quickly and accurately.
Okay. And is the practice at a easy difficulty level, or is it kind of?
It's random.
It's random.
We take random trials from the experiment for each participant.
Okay, now Alberto Testolin, who is in this series, he'll be speaking later. Do you have a question as well?
Yeah, thanks. Thanks, Tali, first of all, for the very nice talk. I'm well aware of your work because I work with Marco Zorzi in computational modeling of this kind of processes.
Yeah.
And I really like your approach. So I just wanted to jump in because I saw that Stefan was very interested in this temporal estimation problem for numerosity. And I just want to say that I can include a few slides later in my talk about this, because we are tackling this problem, actually. We are thinking about how to do that. Also taking into account, of course, the continuous magnitudes like frequency, individual length of the event, interval length, tempo, rhythm. It's messy, but we are trying to use a recently proposed stimulus space by the group of De Wind and Elizabeth Brannon, which is trying
Don't get me started.
No, yeah, that's why I will just maybe include a few slides in my talk, and then we can discuss if Tali will join us.
Yes, Tali, you're welcome. As you know, to all of these. I hope you come back.
I'll be happy to. Yeah, thank you.
Thank you, Alberto. Are there other questions from inhibited people? Est-ce qu'il y a d'autres questions? Sinon, I want to thank, if not, then I want to thank Tali very much for her very stimulating talk. And next week, next time.